All NEET aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. In this podcast, molecular basis of inheritance, the subtopic it is regulation of gene expression. So, regulation of gene expression is a broad term, that is an umbrella term. It covers a journey from the DNA to functional protein. So, gene expression is not a single step molecular process, a multi step molecular process. It has to pass through various checkpoints to produce a functional protein. So, whatever the encoded information in the gene, that is the DNA, finally expressed by synthesizing proper protein. So, information to functional protein, a journey, nothing but the gene expression. All the cells are the same DNA. Why does it differ? One gene for neuron, other gene for muscle. That is nothing but gene expression. Gene expression in case of the prokaryote is very simple. Start or stop the transcription level. But gene expression in case of eukaryote is the complex. Occurs, that is exerted at various levels. Transcriptional level, processing level, transport level, translation level. Transcriptional level, this is the first checkpoint, molecular checkpoint. The accessibility of RNA polymerase in a given promoter site regulated by the accessory protein, which affect RNA polymerase in detecting the start site, that is structural gene. It has to pass through that checkpoint. Second, after formation of the RNA, mRNA, that is heterogeneous RNA, it should undergo processing, that is splicing, capping, and tailing. So it should be the processing, that is alternate splicing, is time specific, tissue specific, species specific. Right protein has to produce means it should undergo in a proper way, that is alternate splicing. It has to pass through the second checkpoint. Third, transport from nucleus to cytoplasm. So properly processed RNA only leaves the nucleus, enter into cytoplasm with a cap at 5 end and poly A tail at third end. Then only it leaves the nucleus and enter into cytoplasm. Translation level, the fourth one. The formation of the protein is not going to be a complete process. Post-translational modification occur depends upon the requirement in case of the target sites. That is post-translational modification of the protein. Next, the gene it meant for one specific function or set of functions. If we take the E. coli, Escherichia coli, it synthesizes beta galactosidase enzyme B-gol, which catalyzes the hydrolysis of the lactose into glucose and galactose. Lactose is a disaccharide that hydrolyzed into glucose and galactose by beta galactosidase. And the glucose and galactose used as a source of energy by E. coli. If the medium or environment is without lactose, there is no entry of lactose into the bacterial cell. So there is no necessary to produce beta galactosidase to hydrolyze lactose into glucose and galactose. So here the microbe, the prokaryote utilizes energy efficiently. Whenever there is a lactose, gene switched on. Whenever there is no lactose, gene switched off. So the regulation of the gene, it is metabolic, environmental, physiological. Whenever it is required, gene switched on. Whenever it is not required, gene switched off. So it is a regulation at metabolic, environmental as well as physiological. Next, even the embryo differentiate and develop into an adult organism is controlled and coordinated by different sets of the gene. If there is no coordination between different sets of the gene, there is an anomaly in case of the organs, in case of the adult human being. So there must be some coordination between different sets of the gene. Next, in case of the prokaryote, earlier we said it is very simple. Start or stop the transcription. Rate of control of transcriptional initiation itself a predominant target site for control of the gene expression in case of prokaryote. Because in bacteria, transcription, translation are coupled. That means it occurs in a single compartment. No demarcation of nucleoplasm and cytoplasm because it is without nuclear membrane. Further, RNA do not needs any processing because the genes are non-split. So, in case of the prokaryote, the gene expression is very simple. That is speed and efficiency in case of prokaryote. But in case of eukaryotes are complex organism controlled with the precision required in case of eukaryote. Next. So in case of the prokaryote, in case of E. coli, if you take the example, RNA polymerase at a given promoter in a transcription unit is regulated by interaction with accessory protein. So accessibility of RNA polymerase at a given promoter in a transcription unit regulated by accessory protein. This accessory protein enable or allow the RNA polymerase to detect or to prevent from start site. That means the protein, accessory protein act positively, that is activators, allow RNA polymerase to bind at promoter to detect start site. It is repressor, it do not allow RNA polymerase to access to promoter and unable to detect start site. So accessory protein works positively as activators, negatively as repressors. Next, the access of the promoter to RNA polymerase in prokaryote in many cases regulated by interaction of the operator with accessory protein. That means the operator is adjacent to the promoter. In many of the operon, the operator receives the biochemical or protein of the I gene, that is regulator gene. Once this protein blocks the operator, there is no accessibility of RNA polymerase to promoter. So, in case of operon, an operating system, that is a group of genes operating together for a common function, what we call operon. Operon includes promoter, operator, regulator, and structural gene. So each operon having its operator as well as repressor. In case of suppose the example lac operon having its own lac operator as well as lac repressor. Lac operon, the first transcriptionally regulated gene expression by interaction of the two scientists, a geneticist Francois Jacob and a biochemist Jacques Monod. The contribution of these two scientists led to the elucidation of transcriptionally regulated gene expression in case of E. coli. This is a polycystonic gene expression having many structural genes. So in this lac operon, it is having the I gene, that is regulatory gene, and promoter gene, operator gene, and three structural genes. All constitute an operon. An operon is an operating system for a common function, that is lactose metabolism, that is lactose catabolism. So lac operon is a prototype gene expression. The prototype is initial model system to understand how genes are regulated, how genes are producing the product. So to understand the gene regulation. Later several models are expressed, like it is trap operon, tryptophan operon, val operon, valine operon, his operon, histidine operon, era operon, arabinose operon. All operons are there. So now we are concerned with the lac operon. It is a polycystonic structural gene in case of E. coli. Next. So this lac operon having how many genes? That is I, that is inhibitor, not it is an inducer, it is a regulator gene. and it is promoter gene, operator gene, and 3 structural genes ZYA. So total number of genes it is the 6. Number of types of genes: I gene, promoter, operator, and structural gene. 4 types of gene. Total number of genes 6. Number of type of genes 4. And number of structural genes 3 ZYA. Next, the I gene produce repressor mRNA that produce repressor protein. Next, P promoter where RNA polymerase binds initiate transcription. O operator gene which receives the biochemical produced by the I gene, that is regulator gene, that is repressor protein. And structural gene ZYA, Z gene code for beta galactosidase, that is BGAL, that hydrolyze beta galactosides, that is lactose into glucose and galactose. Y gene, it code for permease, which increase the permeability of lactose from medium to the cell. And A gene, it code for trans-acetylase. So no direct role in case of lactose catabolism, but it is an accessory protein. During metabolism, many byproducts are formed. Some of the byproducts, it may be toxic. The toxicity is detoxified by the addition of acetyl group to those toxic substances by trans-acetylase. It adds acetyl group. So overall, all these 3 gene product, Z gene, Y gene, A products are useful for lactose metabolism, that is catabolism of lactose. Even in other operons, it works in the similar way or related metabolic pathways. In this pathway, it is catabolic. In other pathways, it may be anabolic pathways. Next, lactose is not the part of lac operon, but it acts as a inducer. It is responsible for switching on and off. Whenever there is a presence of lactose, genes switched on. Whenever there is absence of lactose, genes switched off. So lactose, it is an inducer. So lac operon, it is inducible system. The inducer, it is lactose. Next, the preferred substrate is the glucose. Why it is a preferred substrate? The catabolism of glucose is easy and it provides efficient energy, immediate source of energy. In absence of the glucose, in growth medium, lactose is provided. Lactose enters into the cell due to activity of permease. So permease is a membrane bound protein or enzyme responsible for transport of lactose from medium to the cell. We can frame a question here. How many types of enzymes are present in cytoplasm and membrane bound? In cytoplasm, beta galactosidase, trans-acetylase. In membrane bound, that is permease. So without permease, there is no entry of lactose from medium to the cell. So a low level of lac operon must always be present. Next, the I gene, that is constitutive gene that produce repressor mRNA, that produce repressor protein. So repressor protein always produced by I gene constitutively, regardless of its use, regardless of its lactose presence or absence of lactose. It is a constitutive gene. The constitutive gene product, it is repressor protein. The repressor protein produced always in active state. It is always active. Next, when there is a presence of lactose, the lactose convert into isomeric form yellow lactose. The yellow lactose binds to repressor protein. Repressor protein got inactivated. So repressor protein do not binds to operator. Once repressor protein do not binds to operator, RNA polymerase access to promoter, transcribe ZYAG. In absence of lactose, repressor protein binds to operator. There is a blockage of operator. Even though RNA polymerase binds at promoter, it do not move. There is a blockage by repressor protein at operator. In presence of lactose, lactose converted into yellow lactose binds to repressor protein got inactivated, it do not binds at operator. So RNA polymerase moves as there is no blockage of operator by repressor protein. It moves along operator, transcribe Z, Y, A gene to produce the product. And Z gene, Y gene, A gene, which are three structural gene, each structural gene having start codon, stop codon. Z gene having start codon, stop codon. Y gene having start codon, stop codon. A gene having start codon, stop codon. So each gene product is independent of other gene. Next. So it is a multi-step regulated system, but essentially it can be, that is lac operon regulated by the lactose, can be also visualized as enzyme synthesis by its substrate. The substrate is lactose, raw material it is lactose. For lactose to catabolize, it requires the enzyme. So essentially, lac operon can be visualized as enzyme synthesis by its substrate. The substrate is lactose. It needs the help of beta galactosidase to hydrolyze lactose into glucose and galactose. Next, how long lac operon will occur in presence of lactose? Till glucose level increases or lactose decreases. So whatever the products formed during breakdown of lactose, glucose and galactose never used as inducers. Only lactose is a inducer. Real inducer it is yellow lactose. Next. In lac operon, lac repressor in lac operon, regulation of lac operon by lac repressor is a negative regulation. It is a negative, but it is inducible. In presence of it is lactose. So, lac operon by lac repressor is a negative regulation, but it is also under positive regulation too. That is adaptation by the E. coli. If there is no glucose available, it also having a system to catabolize this lactose in some other way to make use of for it is energy by hydrolyzing the lactose. So it can be also visualized as positive regulation too, because the repressor regulation it is negative. So negative, but it is inducible. Negative because of repressor, inducible because of that is lactose. I am signing off from this episode with the completion of this molecular basis of inheritance. In case of the following episode, we will discuss the questionnaire with regard to this molecular basis of inheritance. Very, very important in NEET point of view, the tricky questions and concept based question and statement wise question. So don't forget to subscribe and share to other person, those who are preparing for the NEET with a seriousness.